Hello, everyone. My name is Darla Saunders, and on behalf of the Atlantic Salmon Conservation Foundation, I would like to welcome you to today's webinar. This series is made possible in part thanks to the contributions of the Government of Canada. We're delighted today to be welcoming Dr. Ian Bradbury. Dr. Bradbury is a research scientist with the Department of Fisheries and Oceans and is the Cox Fisheries Scientist in Residence at Dalhousie University. Originally from Newfoundland, he completed his PhD in 2007 at Dalhousie University and started with DFO in 2010. His research uses genomic tools to inform the conservation and management of marine and anadromous species from throughout North America. After the presentation, we'll be opening the floor up for a question and answer session. You'll have the option of asking your questions directly using your microphone, or you can type them in and we'll read them aloud. I will now turn the webinar over to Dr. Bradbury.
Hi, yeah, thank you. I am having a little bit of trouble with the audio, so if I cut in and out, please let us know. So what I would like to talk about today is work that we've been doing on sort of the conservation, genomics, and genetics of Atlantic salmon. And the goal here is really to capitalize on new technologies, new genetic and genomic technologies to help inform salmon conservation and management. A lot of the work I'll talk about is really work that's been done by graduate students and postdocs in my lab, and I'll try and highlight those contributions as I go along. So just a little bit of background. I suspect a lot of us are quite familiar with Atlantic salmon, but they're widely distributed across the North Atlantic. Life histories are characterized by long distance migration. We have individuals moving to feeding areas in the north, such as off Greenland, yet we have high rates of natal site fidelity and natal homing. In conjunction with this, we see large variation in life history across areas, across habitats, and even within some regions. An interesting fact about Atlantic salmon is that currently there are many more individuals in culture than there are in the wild, and that's something I'll come back to later. The current range of Atlantic salmon has been changing over the last tens of thousands of years. The white here represents the extent of glacial coverage during the last glacial maxima, and it just gives an indication of how much the range really has changed in the last 20,000 years. So during glacial maxima, Atlantic salmon were really relegated to glacial refuges to the south, both in the east and in the west Atlantic. Following glacial retreat, salmon colonized northern regions in both the east and west, and we currently see this. The black lines here are a good indication of the what we think the ocean ranges of Atlantic salmon, and in both the east and west, salmon colonized freshwater habitats northward. Despite this, we did, given these large periods of isolation during periods of glaciation, there is significant divergence between salmon, genetic divergence between salmon in the east and west. And we've estimated that salmon in the east and west have around 600,000 years of isolation. And in fact, the divergence can be so great that we even see different numbers of chromosomes or different karyotypes. And that's thought to be associated with this presence in these different refugia for extensive periods of time. So, as a lot of us are aware, across the North Atlantic now, stopping declines in many places. On the left here are just some headlines from the last year in Eastern North America. We have declines being reported in Newfoundland, in Atlantic Canada, as well as in the US. And these kind of match the longer-term trends we're seeing in many regions from the IC stock assessments, which are the data shown here on the right. In many regions, we've seen longer-term trends of decline. And in some regions, such as in Newfoundland, we've seen declines that have been more prevalent in the last couple of years. But nonetheless, there are generally concerns regarding long-term declines across the North Atlantic and trying to understand what the causes and threats to Atlantic salmon could be. So to that end, in my lab, we think a lot about what these potential threats and stressors could be, and we work towards trying to understand them. So one that we think a lot about is interactions with salmon aquaculture, particularly escaped farm salmon. These are just two reports of salmon escape events from last year, one from the west coast of Canada and the other from Scotland. And these are not infrequent events. As these salmon escape, of course, there are potential genetic interactions, both direct and indirect genetic interactions, an example being interbreeding, that have the potential to change salmon populations at the genetic level and alter their life histories or biology, and ultimately their productivity and perhaps challenge their persistence. The other threat that we think a lot about are interactions with fisheries, so fishery exploitation. Now, for the most part, fisheries have ceased for Atlantic salmon at sea, but there are still a couple of locations where salmon are harvested at sea, and there are ongoing concerns regarding interception, particularly of threatened or endangered populations to the south, of populations as they move to northern feeding areas or along migratory routes. And the fisheries that we work on are things like the fishery in the French waters around St. Pierre and Miquelon, off the coast of Labrador, or off the coast of Greenland. So what I'd like to do in this talk then is sort of show how we are using genetic and genomic tools both to understand the resource that is Atlantic salmon, the possible threats that might be impacting it, and how we can use this to inform conservation and management. So the four sort of things that I will talk about are things like the consideration of biocomplexity and the design of conservation units, trying to understand what we have in terms of stock structure, using this data to understand fishery-associated exploitation and mortality, using genetic tools to understand the impacts of salmon, aquaculture, on wild salmon, and really focusing on the direct genetic impacts. And finally, some work that we've just started doing, and this is preliminary, but I think it'll be interesting to a lot of folks, is some work we've been doing on using genetic data to reconstruct trends in effective population size across the range and look at possible correlates that might give us some understanding as to what factors are associated with decline. So I know I've been saying genetic and genomic a lot. When I say that, for those who are not as familiar with the term, I'm essentially meaning that we're using DNA. So DNA contains the full set of instructions for building an organism. It's contained in the nucleus of almost every cell. It's written in this four-letter language, A, C, T, and G, and we can think of it as sort of a book analogy where the salmon genome, which contains about 3 billion of these letters, is divided into chapters or chromosomes, and we have either 27 or 29, depending on where we are, perhaps. And each of those chromosomes or chapters is subdivided into sentences or genes, and these genes code for proteins, which are essentially the building blocks of multicellular organisms. The salmon genome contains around 20,000 genes in total. So what's really nice then is that the genome contains a complete record of an organism or a species ancestry, its history, as well as how it's adapted to its local environment. And this gives us really unprecedented power to identify individuals, populations, and functional genetic variation. And all that means is understanding what makes these things unique, what makes individuals unique, and what makes populations unique. And there's been some dramatic changes in the past trying to unlock all this information contained in the genome has been really impossible. But in the last decade or so, we've seen dramatic changes in our ability to read DNA that have really opened up this resource to not just those working on human genomics, but also those of us working on non-model species. And the main reason for this is that this technological advance in how we read DNA. So this is just a plot from the National Institute of Health in the US. That just shows the cost of sequencing a human genome and how that's changed over the years. So in around 2001, it was over $100 million to sequence a human genome. And that, for a long time, that tracked, this is a line which represents the increase in computing power over time. So that tracked how technology in other fields was increasing. And then around 2008, we saw this dramatic change in actually how we read DNA, to the point where in the last couple of years, we are getting close to, if we're not there already, to a $1,000 genome. So pretty substantial and incredible advance in technology that's really opened up opportunities for those of us who want to capitalize on that for sort of applied fisheries conservation and management. So what we have in terms of Atlantic salmon and stock structure and management units, this is not something that, this is something that's been thought about for a long time, and there's been a lot of previous efforts going in to try and understand population structure and then translating that into management units, or from the COSEWIC perspective into DUs. So COSEWIC took a stab at this in 2010 with their Atlantic salmon update. They identified 16 designatable units spread across the range, the North American range, and those are shown here by these boxes. It was recognized at the time, however, that a lot of these DUs were probably large and should probably be subdivided, but at the time, sufficient data to redesign many of these did not exist. And examples that jump out at us are things like Labrador, where we have the entire region as one designatable unit. So when I started with the department in 2010, one of the first things I started working on was trying to build a better picture of stock structure and population structure for Atlantic salmon in eastern North America. And that, at the beginning, that really involved flying into places that look like this and collecting juvenile salmon. So we would fly in, we'd land in suitable juvenile habitat that looks like this, and we would electrofish, collect juveniles, take a small piece of tissue back to the lab, where we would analyze a targeted set of markers to try and build a picture of how salmon in different regions are related and what population structure is. So in the lab, we've been using a variety of tools to identify population structure in salmon over the 8 years since I've been with the department. Initially, we started with microsatellites, but in the last couple of years, we've switched to single nucleotide polymorphisms, or SNPs. Using a targeted SNP panel, we've been able to develop a range-wide baseline for Atlantic salmon, and that's shown here in the map. And this is really the work of a postdoc working with me by the name of Nick Jeffrey. So this baseline contains around 300 populations, around 7,200 individuals, and it's for 96 targeted SNPs. And so the fact that we've got a 96 SNP panel means that this is something we can genotype, analyze really quickly and cost effectively, which is great for a management tool. Using this panel, we can identify 27 regional groups right across the range. So this panel was really designed to maximize resolution within Eastern North America. And that's why we have a lot more groups that we can identify off the coast of Canada and the US. So we have around 20 groups in North America that we can identify, and those are indicated here by the different colors. And then in Europe and Iceland, we have another 7 groups, including a group in West Greenland. So using this panel, then we can get around 90% or higher accuracy of assigning individuals to these groups. And just zooming in here, this shows what the North American groups look like. So one thing that's obvious right away is that we clearly see this subdivision in Labrador that was not represented in the COSEWIC DU structure. And that's this isolation of Lake Melville, or divergence of Lake Melville in comparison to coastal populations. And that's something that comes up again later in the talk. So in addition to these regional baselines, there are often instances where we want really fine scale resolution in terms of identifying individuals. We may want river scale assignment for fine scale fishery analysis. So in Labrador, using that SNP baseline that I just showed, we might have had 2 or 3 groups that we can identify. We've been exploring other approaches to really increase our assignment power. And one of them is using, sort of revisiting an older technology, which is microsatellites, but really taking advantage of these new advances in how we read DNA, how we sequence DNA. So in this case, rather than doing 5 or 10 microsatellites like we might have done a decade ago, we're sequencing 101 microsatellites distributed across the genome, and that really gives us the ability to measure a lot more alleles, just to measure a lot more things, and to have a lot more power in terms of identifying populations and assigning individuals. So for example, this is our work in Labrador here on the right. As I mentioned previously, we could identify maybe 2 or 3 groups in Labrador. Each of these colors represents a location or group of locations that we can identify. So across these 36 locations, we can identify 26 individual rivers or pairs of rivers. So that's an order of magnitude increase in our assignment ability, and we're almost, for many regions, for many rivers, we are to river of origin, but for other places, especially rivers that might share an estuary or are very close, then we lump them together. But still dramatic increases in our assignment accuracy for identifying salmon populations. And this is something that we're currently extending. We're doing work in southern Newfoundland, in Placentia Bay and Fortune Bay, and we're extending this to the Gulf of St. Lawrence as well. So as we build these pictures of stock structure and population structure across the range and in North America, one of the things that we're learning a lot about are what forces structure and influence population structure. And two of them that are perhaps most prevalent are things like local adaptation, and that's how populations may be adapted perhaps to climate, and how climate may drive population structure. And these relationships are critical for understanding not only the relevance and the mechanisms driving it, but also for predicting responses in the future, and perhaps predicting responses to climate change and range shifts. Also, colonization history, as I mentioned, the range of Atlantic salmon has changed dramatically over the last 20,000 years. And with recolonization of northern areas, we see the potential for recolonization from different places and for regions of secondary contact to become established, and for those to be important in terms of contemporary population structure. So in terms of adaptive diversity, we've done a little bit of work on this, and this is again is a paper by Nick Jeffrey that we published last year. We were able to identify here sort of range-wide clinal variation that we see on both, and when we're using the dots are colored by the genetic group we see, and we have two genetic groups. We have this cline, so we see this nice coherence between clinal variation in the genetics and clinal variation in climate. And in fact, we see this in the East Atlantic as well, and it's something that we might expect, but it's nice to be able to see it. On finer spatial scales, we're also seeing temperature perhaps consistent with adaptive diversity and adaptive differences. This is some work done by Emma Sylvester, who just completed her master's at Dal last year. Here, she was looking at population structuring within coastal Labrador and within Lake Melville. And again, that main signal that she detected in terms of population structuring was really this isolation of Lake Melville itself from the coastal populations. Now, Emma's environmental analysis very clearly showed that there was a strong correlation between this population structure and temperature, but it wasn't sort of cold temperatures like we were seeing in the clinal analysis. In this case, it was maximum or maximum summer temperature, suggesting that this break between the coast and Lake Melville was really being driven by a difference between sort of the coastal climate along this region and a more continental climate with much warmer summer temperatures inland. And again, that's consistent with sort of adaptive diversity driving population structure in this region. So in terms of colonization history, we've also been looking extensively at trying to understand how colonization history has helped shape the salmon populations we currently have. So this is work by a postdoc of mine, Sarah Lenhart, working in the lab. Here, we had known for a little while that there was evidence of European genes, if you will, or signatures of European colonization in regions of Newfoundland and perhaps in Labrador. What Sarah's work has shown quite nicely is despite this, we've known we've had isolation of the East and West Atlantic, or they've been isolated for 500,000 or 600,000 years, but it looks like during the last deglaciation, we've had colonization of northern areas, not just to a couple of places, but it seems to be extensive, particularly in the north. So this is focusing in on a region of the genome that tends to be diagnostic for East and West. This is a translocation that occurs between chromosome 1 and 23. We see evidence of that in Labrador as well as into Newfoundland. So this would be the ancestral European type, and we see it sort of a clinal pattern down through Newfoundland. And this is clearly consistent with post-glacial colonization of Labrador by both North American and European salmon somewhere in the last 20,000 years. So all of this information on adaptive diversity and population structure and our understanding of how that's formed through colonization routes, that all feeds into how we might better understand and better define conservation units and from the COSEBIC perspective, designatable units. And this is something we're currently working on in projects in conjunction with other researchers at Dalhousie. Low-hanging fruit, again, are things like the Lake Melville situation. Clearly, I think that meets the criteria of a designatable unit. That will be up for COSEBIC to decide, but it certainly seems to be consistent with that definition. And there are other places, perhaps in Newfoundland, where subdivision could be warranted. And probably elsewhere, as we're currently building up more data for other regions, such as the Gulf, and as we get more data, potential for splitting and subdividing, and more importantly, more accurately representing the biocomplexity that's there exists. So from a management perspective, then, at least immediate management perspective, we can then start to use this information on stock structure to assign individuals caught at sea back to their region of origin. And that helps answer the question, who's catching whose fish and perhaps how many. So the lines here represent what we think are the typical migration routes for Atlantic salmon in the North Atlantic. So we have salmon from throughout Atlantic Canada, moving to the Labrador Sea, perhaps at least the grilse, and then multi-sea winter individuals maybe feeding in the waters off West Greenland before making their return migration. So at present, there are several harvests that might intercept and exploit salmon from both North America and Europe along this route. So in the waters off West Greenland, we have the potential interception of both North American primarily, but also some European individuals, probably coming from the UK or Southern Europe. And then in the Labrador harvest, as well as in the St. Pierre-Miquelon harvest, there are potential for interceptions of fish potentially moving northward or returning. So we've been working on the 3 of these fisheries for, well, since I started, but we have samples that predate me. So for each of these, we probably have a decade or more of annual samples from the harvest that we've been conducting genetic mixture analysis with or individual assignment analysis with to see where these individuals are coming from. And I'll just quickly run through each of these fisheries just to give folks an idea of what we've been seeing. In the West Greenland harvest, this is primarily targeting these multi-sea winter individuals, both from North America and Europe, but primarily from North America at the moment. The harvest in 2017 was around 27,000 tons based on the ICES report. And because there's this potential for interception of potentially threatened or endangered stocks, there's always concern regarding trying to understand what's being taken in this harvest. The analysis we've been doing this for multiple years, and the picture is pretty consistent over the recent years. We see in essence the large multi-sea winter stocks being represented. So Labrador is about 20% of the harvest, Gas Peninsula is around 28%, and the Gulf of St. Lawrence is around 29%, with some minor contributions from other areas. The Labrador FSC fishery is a very different pattern. This is a subsistence fishery that harvests around 10 to 15,000 individuals a year. Most of the harvest occurs within rivers or estuaries, and this is probably part of the reason. This probably functions to reduce significant numbers of interceptions. So for the most part, this fishery is entirely Labrador in origin. 96% or more of the fish harvested assigned back to Labrador. There are a couple of exceptions, and every now and then, every couple of years, we see a US fish or two, but for the most part, this is a local targeted fishery. Finally, the St. Pierre-Miquelon fishery. This is an interesting fishery. So there are no salmon populations on the islands themselves, and this fishery takes place in the French territorial waters around St. Pierre-Miquelon. It's a professional and recreational gillnet fishery, and the harvest has been up to 4 or 5 tons in recent years, although last year was around 2.8 tons. Again, given the proximity to regions or stocks that might be of management concern, like the Southern Newfoundland designatable unit, which was classified as threatened by COSEWIC, there are concerns regarding interception. So again, we've been looking at this fishery since 2004. And again, this gives us a general flavor as to what the stock composition in that harvest has looked like over that time period. 24% usually from Newfoundland, 27% from Gas Peninsula, and 37% from the Gulf of St. Lawrence. So obviously we can, and this is something we continue with every year. We'll continue to get samples from these fisheries, and that feeds directly into both stock assessments done within the department, as well as international stock assessments done by ICES. But in addition to trying to understand stock composition in fisheries harvests, we can also use this sort of tool and these assignments to really learn something about migratory routes and timings. So any samples that have been collected at sea of Atlantic salmon over the years can now be analyzed, and we can analyze scales and individuals back to one of these regions and try to understand where fish are at sea and when we're seeing them. So I have 2 samples that we've analyzed so far that I can talk about. One is from the Labrador Sea, taken in the late 1980s, and the other is from the Faroese fishery, taken in the early 1990s. And what's fascinating about these samples is that both of these samples show much more transatlantic migration than we might have expected. So for the Labrador Sea, we see this was a sample of both smolts and adults from 1987 and 1988. And across the board here, 22% of these individuals are identified as European. And when we look at where in Europe, they seem to be predominantly Scottish fish. So what's fascinating here is that we've got smolts that are leaving Scotland in the spring that are making it to the Labrador Sea by the time of this survey, which was late summer. So that's an incredible feat of migration. Interestingly, somewhat similar story in the Faroese fishery. Here, within large salmon, so these are multi-sea winter fish, 22% are North American in origin. And when we look at where they're mostly from sort of the Newfoundland, Gas Peninsula, Gulf region. So it's very similar to what we see in St. Pierre. It's that group in the southern portion of Atlantic Canada. So then in both cases here, we're seeing more evidence of, potentially more evidence of transatlantic migration than we might have thought previously. So we're currently building on that, and this is an analysis that's currently underway. I don't have any results yet, but I can at least show the distribution of the samples. These are scale samples that we've kind of scoured the DFO holdings going back to the 1960s, and we've pulled out any samples of scales collected from Atlantic salmon at sea. And so the points here represent samples that we're currently analyzing from fish collected at sea from the 1960s to the 2000s. So for each of these fish, we will get region of origin to one of those 27 or 28 reporting groups across the range. So hopefully we'll learn a lot about where fish are at different times of the year and where they're coming from at different places here in the offshore area, both from the waters closer to Greenland, but also in the Labrador Sea and even a few on the Grand Banks. So shifting a little bit, the second kind of threat or stress that we've been focusing on then is this idea of aquaculture impacts, and particularly direct genetic impacts on wild populations. Where we work in southern Newfoundland, we regularly see escapees such as this one, which are showing up at our counting fences around the south coast, either the Garnish River, primarily the Garnish River. And it can be particularly worrisome when we see individuals showing up like this, which we saw in the fall of 2013. Here, escapees are showing up in spawning condition in the fall of the year. So when we see that, then it's highly likely we will have interbreeding and then have genetic interactions. And this sort of thing has been noted elsewhere. It's been noticed in the UK, where it's being called the invasion of Viking salmon. Here's a headline saying that 1 in 4 salmon in the UK are hybrids. And of course, work we've done here in Atlantic Canada is showing similar things. With interbreeding, we expect genetic changes in these populations. There's been work done in Norway recently, which is showing how the life history of Atlantic salmon there, of wild salmon, are changing due to interbreeding with farm escapes. And we've seen similar genetic changes in North American populations. So it's clear that escapees that farm escape salmon can interbreed and are changing wild populations in many regions. So in Norway, some recent work suggests that as many as 150 populations across the country show genetic impacts, and this can be as high as 47% of a population's genetic makeup being attributed to interbreeding with escapes. There is evidence from North America and the UK, as I mentioned, although study has been limited. And it's been limited for a variety of reasons. It can be difficult when you've got a diversity of stocks within the aquaculture line to quantify these impacts. It can be difficult when you have limited pre-impact baselines. And what's been lacking in a lot of instances is really what's been needed is sort of a single escape event where you've got sufficient power to really quantify the geographic scale and the magnitude of these impacts. And such a study could really be used to inform impact management, mitigation, and siting decisions. So we had an opportunity in 2013 to study such an escape event. This is one that's been quite well publicized. In 2013, there was an escape of around, by all accounts, around 20,000 market-sized mature salmon into the south coast of Newfoundland. So the escape event happened right here. This is the island of Newfoundland. The escape event happened right here. Our best guess as to the entire wild population for the south coast is around 20,000, give or take, wild salmon. So given such a sizable influx of farm salmon to a very localized area, we were really probably expecting to see some sort of impacts. So it's also Worth noting that this escape happened in September, just prior to spawning, and we were seeing fish showing up in spawning condition, escape farm salmon showing up in spawning condition shortly after. It's also worth noting that the dominant aquaculture strain used in the area at that time was from the St. John River, so there was a lot of genetic divergence between that strain and wild populations, and it was into a region that was relatively pristine. So we really had a lot going for us in terms of quantifying these impacts. So the goal then was to try and quantify impacts following a single escape event in North America and use it to explore the spatial and temporal variation in hybridization. So following that escape event, the very next summer, we set out to sample young of the year. And again, that meant flying into a lot of places along southern Newfoundland, electrofishing, taking young of the year salmon and taking a small fin clip, bringing the fin clip back to the lab for genetic analysis. So in the lab, we had designed a series of genetic panels that are collectively diagnostic for escape farm salmon and have enough power to identify hybrids, either hybrids or hybrid class when they occur. This just shows the distribution of the markers across the genome. They're widespread across the genome. The red ones here are some of the markers in one of our 96 SNP panels. And we've done a lot of work. Again, this is work done with Brendan Ring when he was a postdoc in my lab. A lot of work on developing statistical approaches and analytical pipelines that we can use to design these panels, to quantify their potential accuracy, and power in terms of identifying hybrids and hybrid class. And then one example is this R package that Brendan developed called Hybrid Detective. So this is just an example showing how the overall performance of some of these panels changes with our sort of degree of certainty, how certain we want to be, and for different panel sizes, which are the different color lines. So we have a plot for pure farm, for pure wild, and hybrid, and we can see that for panels of 96 SNPs or more, we have high accuracy for identifying these three groups at different levels of posterior probability. So clearly we have enough power to identify hybrids and hybrid class when they occur, and we use this to quantify the impacts following this escape event. So again, this is where the escape event happened. These are the locations we surveyed, and overall we saw evidence of significant hybridization following this escape event. 27% on average of the young of the year samples had one or two aquaculture fish as a parent. Hybrids were detected in most rivers, 17 out of 18 rivers sampled, and feral offspring, so individuals which had two aquaculture offspring, parents, were detected in 13 rivers. By far the most common hybrid class detected were F1s, so first generation hybrids, but we did see evidence of some other hybrid classes, so F2s and backcrosses, and that suggested that there had been prior escape events that really had not been examined. So using this, this presented a really good opportunity then to try and focus on what factors influence the distribution of hybridization and how the levels of hybridization may change over time to try and estimate the strength of selection against hybrids in the wild. So we first started out looking at what factors influence the distribution of hybrids in the field following this escape event. And the first thing we saw was that population size, wild population size, seemed to be really important. Now, we don't have great data on wild population size for many of these systems, but we do have a couple of proxies that correlate well with wild population size. So two of them that we've used is river axial distance and sort of the mean salmon angle per year in these rivers. And we've shown for a subset of other rivers where we do have count data that these quite nicely reflect wild population size. So what we see clearly here that there are associations between river or population size and the proportion of wild, feral, or hybrids that we detect. So as rivers get bigger and populations get bigger, we have a higher proportion of wild individuals encountered. And similarly, in larger rivers, we have much fewer feral or fewer hybrids detected. And it suggests then that larger rivers are more robust or potentially resilient to escape events. Within river systems, we've also done some extensive sampling within some of our larger systems to look at what landscape features might be associated with levels of hybridization. And two of them that jump out here are things that essentially are associated with the amount of work or effort it takes escape farm salmon to enter these systems. So here are the proportion of pure farmed, hybrid, or wild salmon moving in or found in these systems. And within river systems, we see that if we increase essentially the work it takes an escaped farm salmon to move into these systems, we see the number of wild young of the year encountered goes way up, and the number of feral or hybrid go way down. So the amount of work it takes is a significant predictor of proportion of hybrids encountered. But also, we also see a pattern with number of partial obstructions. So as we see more partial obstructions, again, we see more wild fish and fewer hybrids or farm fish, feral offspring. So the conclusion really is that smaller populations are more at risk, and escaped farm salmon are much less capable when challenged by migratory obstacles. So areas of rivers below, closer to the ocean, or below migratory obstacles would also be more at risk. So as I mentioned, we also use this to examine temporal changes in levels of hybridization. And this was a great opportunity to sort of follow this 2014 cohort through time to see how they survive compared to wild individuals in the field. So we went back in 2015 and in 2016, sampling these habitats and then targeting sort of 1+ and 2+ individuals so that we could track changes over time. Each of these lines represents a different river. The bars here represent the mean and standard deviation, and we can see clear trends. So in the wild fish, as we go from young of the year to 1+ to 2+, the proportion of wild fish increases dramatically. We see almost every river, we see dramatic increases in the proportion of wild fish moving with that cohort over time. Similarly, the proportion of farmed fish, or these feral offspring, it plummets. We see decreases in almost every river to the point where in many systems they've disappeared. Hybrids is a bit noisier, but we do see a general decrease over time. The only exception are the backcross wild, where we see some evidence of an increase. And that perhaps is expected because these would be most similar to the wild individuals. So we've used this to estimate sort of the strengths of selection or the relative survival of F1 and feral individuals compared to the wild. And we get around, for feral, we get around 0.2 relative to the wild for their survival. For hybrids, we get around 0.8. So it clearly suggests decreased survival of feral and hybrid individuals relative to wild individuals over time. And that's what we'd expect. They likely deviate from wild individuals in terms of their some of their traits, growth rates, behaviors, and things like that. So then what does this mean for population persistence and stability? We would expect that, given that these F1s are not selected against entirely, that some of them do survive, we'd expect changes in these populations. And evolutionary theory would predict that as we see deviations from sort of the desired or the optimal for these populations, that we would see reductions in abundance. And as I mentioned earlier, we are seeing changes in populations in other places due to interbreeding with escapees. And of course, we're seeing evidence of decline in some places. So there is data that's consistent with these predictions. But what we wanted to do was to sort of take that a little step further. And we've done some modeling work. So this is some individual-based genetic modeling or ecogenetic modeling, which incorporates our realistic estimates of relative survival of feral fish and hybrids into a salmon life history model that allow us to try and understand at least what the scale of potential outcomes could be. So in this case, we're using the ecogenetic model produced by Marco Castellani and colleagues in 2015, and we're incorporating our realistic estimates of survival and sort of based our environmental and population characteristics on Newfoundland populations, on southern Newfoundland populations, drawing a lot of, or some of our information from things like the Conn River or the Garnish River. So these are just some example runs that I've included. We've started with a wild population size of 500 individuals. For each plot, we have 2 levels of invasion, and we've spread out the invasion. So here we have continuous invasions over 10 years, over 50 years, or over 100 years. And a couple of things that are worth noting. First of all, that with high levels of invasion, where you've got invasions that are equal to or greater than the wild population, we can see significant declines that happen very quickly. When you lessen the size of the invasion, then we lessen the size of the decline, to the point where when you're in 10 to 20%, there's little demographic impact detectable. The other thing that's noteworthy is that when invasion stops, even with significant intrusion, we do see recovery, and recovery can happen over 50 to 100 years. Of course, from a practical perspective, stopping invasions can be perhaps unrealistic. So to summarize the aquaculture stuff, this great opportunity really to study this single escape event in a relatively pristine environment has really shown us that Hybridization following a large escape event can be widespread, and it can account for a significant component of production the year following. Hybrids were present extensively across the area, and there were some landscape correlations that were worth noting. Things like smaller rivers had more hybrids, and more accessible locations had more hybrids. Following the cohort over time has really shown us that feral and hybrid individuals do have reduced survival compared to wild, and the proportion of wild individuals increases over time in most rivers. However, for the hybrids, particularly the F1s, they do still survive. So there is still the potential for interbreeding, particularly as precocial males, and for changing populations. And the individual-based model predictions clearly show a range of outcomes, but I think it's worth noting that under high invasion scenarios that population declines certainly are predicted from these sorts of simulations. So the last thing I want to talk about is some work we've been doing more recently. This is, again, work that Sarah has been doing in conjunction with Tony Kess, again, both postdocs in my lab. And here we're trying to use genetic data and genomic data to sort of reconstruct trends in effective population size throughout the North Atlantic over the last few decades and understand what factors might be associated. So this really draws on a new method that was published a couple of years ago that allows us to reconstruct changes in effective population size using genome-wide data. When I say effective population size, this is really just an analog to census size, but it's like an evolutionary complement. So it represents the size of an ideal population that functions in an evolutionary perspective the same as the population being considered. So what we've been doing here is sort of capitalizing on this new method and on our range-wide genetic data and genomic data that we have, where we have geographic locations distributed right across the North Atlantic, but also we have good genomic coverage in this data. So that together allows us to use this method to try and reconstruct trends in effective population size for salmon right across the North Atlantic. And it's really the first time that this has been attempted. So what Sarah and Tony have shown here is that we can detect changes and declines in many populations across the range. Here we're focusing on 1975 to 2005, so a 10-year period, and we've lumped populations. We've compared sort of estimates of effective size at the beginning and start of that time period, and we've identified populations that show significant change spanning that time period. And this is what I mean by significant change. So these ones are ones that would have declined, whereas the blue lines here are ones that were characterized as no decline. We've plotted those here on the maps. I mean, overall, around 63% of North American populations and 61% of European populations show significant changes in effective population size over this time period. So quite remarkable that we're getting a similar number in the East and West. And of course, a lot of variation in some places with neighboring populations showing different trends. So it's really important to try and understand what factors might be influencing these changes in effective size. The other thing I can say is that we have a couple locations, 3 locations, where we have older samples and newer samples. So we're not doing a reconstruction, we're actually comparing changes in effective size using old and new samples. And we get patterns that are exactly consistent with our predictions based on this reconstruction. So it seems to be quite robust. So Sarah has done a lot of environmental association analysis, trying to understand what factors are associated with decline across the range, doing it separately for North America and for Europe. And she's done things like random forest as well as redundancy analysis. And the pattern seems to be consistent no matter what we do. The declines are generally associated with warmer winter temperatures across the range. So as warmer winter temperatures occur, we see a greater tendency for populations to show decline. And that's both in the Northwest and in the Northeast Atlantic. And that's consistent with what we might expect based on overwinter mortality in fresh water. The other thing we've done is look at focus on markers that are associated, specifically with genomic regions that are associated with decline. And if we do that, we can clearly show that there are strong differences among populations that are declining or ones that are not declining. So we've identified around 200 plus SNPs in North America and in Europe that are associated with these declines, and they're spread across the genome. They're not lumped in clear genomic regions. And when we do that, So for North America here, we see clear differences between ones that are not declining in blue versus ones that are declining in red, and the same for Europe. Just on a simple PCA, we can get clear distinction between the two groups. So that's kind of cool that there may be a genetic predisposition under the current regime for decline versus not decline. And when we look at these genomic regions that are associated with decline, we see that they may be associated with areas or traits that have been previously identified in terms of being associated with adaptation or morphological and life history variation. We see QTLs or possibly genes that are associated with metabolism and body size or age and maturity. So some of these things are starting to make sense. But again, this is still early in our analysis of this data. So this is a last slide here I want to show, but this is again preliminary, but we've kind of taken that to the next step. So borrowing from the sort of the human genetics field, where associations with diseases are often made not with single genes, but with multi-genes, and then those can be used to predict clinical outcomes. Here, what we've done is we've developed sort of a polygenic risk score for populations like using a high-density SNP array. That allows us to predict, essentially using the data we already have, if we are likely to see population declines or no decline. And so using this polygenic risk score, we've made some predictions using data we have on hand for North America and for Europe. Again, this is very preliminary, but I think it's a nice illustration of how we can start to use these associations to try and make some inferences in terms of what factors might be involved, and perhaps some predictions about what populations might be likely to experience in the coming years. But again, very preliminary. So to summarize here quickly, a lot of what we've been focusing on are looking at population structuring, looking at regional groups, and we've been doing a good job of resolving regional groups, and more recently working on fine scale structuring and understanding what forces might be driving these populations or population structuring in these systems. We've been using this to understand fishery exploitation, and that continues. We have a couple more years of funding to do that, so we'll continue looking at the composition of these mixed stock harvests, and in the next year we'll try to combine all of these fisheries into a single estimate of exploitation for each of these groups. But what's even more interesting, I think, is using these historic fishery samples or samples collected at sea to try and learn more about migration routes and timing in Atlantic salmon in the North Atlantic. And I think that will be a quite powerful approach to try and better understand where fish are at given times. And finally, for the aquaculture interactions work, we've shown quite nicely that there's interbreeding following large escape events. And what's on our plate now is to understand the biological consequences of this for individuals and for populations. We're developing baselines for new areas like Placentia Bay. We've been out generating a pre-impact baseline for the new expansion or the proposed expansion for Placentia Bay. And we're developing monitoring tools to evaluate the presence of escapees based on eDNA. So with that, I have, of course, an incredibly long list of acknowledgements here of all the grad students and technicians and postdocs and partners, industry, and First Nations partners, as well as government agencies, funding agencies, universities. And with that, I would be happy to take some questions.
Thank you so much. So for participants, you have a couple of options for asking questions. So using your webinar control panel, which is the little gray box you should be seeing in your upper right-hand screen, you can either figuratively raise your hand, which is the little yellow hand icon, and we can unmute you and you can ask your question directly, or you also have the option of typing in your question and we'll read it aloud for you. If your control panel is minimized, if you hit the orange box with the white arrow on it at the top, it will expand again so that you can use either of those options. So we'll give folks a few minutes. Okay, so our first question comes from Tommy Lansari. Hello, Tommy.
Hi. Hello.
Hello.
Thanks, Ian, for the very, very informative presentation. This is more of a clarification, I guess, than a question, but your mixed stock fishery graph that you showed from the Faroes Islands. Am I correct? You said that the 22% of the fisheries is removing North American stock, and then you had a pie chart, and that pie chart kind of just shows that off that 22%, the split between the Gulf region or the Southern Gulf, the Gasp Bay, and there was a third component. Is that correct?
That's exactly correct. Yeah. So again, to clarify, the 22% is of large salmon that were sampled in the Faroese fishery in the early 90s, so 92 and 93, I believe. And the pie just showed the assignments of those fish to North American reporting groups. So we pulled out the North American groups and looked at where they were from, and it's really those southern groups. So Newfoundland, Gulf, and Gasp.
Perfect. Thank you.
Thank you. So our next question comes from Gérard Chaput. He asks or says, "Thank you, Ian. Fascinating work. What would you recommend as a sufficient or appropriate population structure scale for defining conservation groupings, as for example, to be used by COSEWIC, given that the groups defined seem to depend upon the genomic detail examined? For example, continental separation based on a few markers, 15 microsatellites distinguish 16 reporting groups in North America, 96 SNPs give 20 reporting groups, several river-specific groups in Labrador distinguished with large microsatellite panels."
Yeah, so that really the answer to that boils down to what your definition is of the conservation unit or designatable unit. So with COSEWIC, designatable units are defined based on discreteness and on significance. So the populations have to be discrete, perhaps genetic discreteness, but they've also got to show some sort of significance, either independent evolutionary lineages or perhaps adaptive diversity. So when you're thinking about identifying those, keeping those two criteria in mind, I think, is important. So with the, and obviously it can be hierarchical, but for instance, the microsatellite work in Labrador, I'm not sure, based on the microsatellite panels, there's no evidence of significance there. So we clearly have discreteness, but there's no evidence that each of those groups or small groups of rivers should be designatable units at this time. Not to say that if we did extensive analysis of those populations, we wouldn't see evidence of adaptive diversity between those groups. But at the moment, that data does not exist. At the moment, I think the regional scale is where we're getting the best support for both of those criteria being met, both the significance and discreteness. But of course, it is a bit of a game where the more data we have, the more we learn. So in many of these species, as we get genomic data and more genomic data, one of the consequences is that we do split. It does seem that we're splitting more and more.
Thank you. The next question comes from Pedro Nilo, who asks, "What is the oldest documented case of feral gene introgression?"
So just to clarify, what is the oldest case of introgression of farm salmon into wild salmon? Is that?
I think so. Yes, Pedro. If it's something different, if you could type that in, please.
Okay. So I mean, we've had salmon aquaculture in Eastern North America since the late 70s. So that's the sort of time scale that we're looking at. To our knowledge, the documentation of feral offspring following this escape event is somewhat unique. Now, I know that there's been reports of potential feral individuals being identified on the west coast of Canada at one point, but certainly in the native range, this is one of the first times that we're aware of documenting that.
Thank you. The next question comes from Justin Wilson. Justin asks, you suggested that a decrease in fish stocks may be related to warmer temperatures. Why do you feel that warmer temperatures are reducing stock?
So just to be clear now, so the warmer temperatures are associated, warmer freshwater temperatures were associated with the declines that Sarah measured based on the genetic data. We have only focused on freshwater, so obviously marine survival is a huge component of this, and it's really difficult to try and make environmental associations at the stock level with marine conditions because we don't really have a good understanding of where fish are in the ocean yet. So the amount of variation that we can explain in these declines is not huge. We're talking maybe 10% of the variations explained by warmer temperatures, but that's still significant given variation we see. It's probably, I don't work a lot on fish physiology. I don't work any on fish physiology, but from what I've been told, that warmer temperatures may result in reserves being used more quickly and change overwintering survival and things like that. But that's a little bit outside my wheelhouse.
Thank you. The next question comes from Colin Murray, who writes, thank you, very interesting. This is slightly off topic, but how many chromosome pairs do Icelandic salmon have?
Yeah, good question. So as far as I know, I don't know if any Icelandic salmon have been karyotyped. Certainly our data would suggest that we see, it should have the European 27. But again, we see a lot of variation in North America. We don't, to my knowledge, we don't see that variation in Europe, at least in our genomic data, which we think is indicative of karyotype. They seem to be consistent with Europe.
Thank you. Chris Elvidge has the next question. The data on hatchery fish basically giving up on spawning runs and not reaching as far upstream are very interesting. Is there any idea of whether or not amongst wild fish there are differences in fish quality, size, survival, etc., along those same stream gradients? More specifically, do wild fish have some advantage of further migration?
So whether or not there are differences in the wild fish that make it further up, there probably are. I don't think we have looked at that, so we don't know the answer. There are probably differences in terms of size or condition that would allow some fish to make it further in, but that's not something that we've looked at specifically.
Thank you. John Kosick writes, great talk and comprehensive work. For the effective population size information, it looked like you had a US river that effective population size did not decrease. Was there a US river on the map, and if so, which one?
Yeah, so offhand, I don't remember which US river we have there. I can say that because this is effective population size, and this is the typical caveat that I have to give, this may or may not reflect directly abundance in an anadromous individuals moving in and out, because it's more, it does include variation in individual reproductive success, as well as contributions from precocial individuals and potentially gene flow from neighboring systems. So it's a little bit more complicated. Certainly we see rivers in Newfoundland where we see a similar thing. We've had declines in the number of anadromous fish, and we don't see a significant difference over that time period in the effective size change. So it's an interesting thing.
Thank you. And for our final question, we have a follow-up question from Tommy Ansari, who asks, my question is related to decline in effective population size and the relationship to winter warming. In our area, in East Coast, North America, we still have winter water temperatures near zero. So with winter warming, do you mean that winter duration is becoming shorter and will negatively affect winter survival, or is the correlate the air temperature?
So so I should have clarified. We did use air temperature for this analysis, and of course it's been shown to be associated with water temperature, how that correlates in the winter. But in our analysis, we used minimum maximum winter temperature. But again, it's based on air temperatures.
Great, thank you very much.